Explain mechanistically, you talked about immune suppression and how immune suppression rolls into cancer formation. Explain mechanistically how that works. And are there other facets of the mRNA vaccine and the other types of vaccines that could be coming into play in terms of cancer formation?
Okay, yeah, this is an excellent question. So certainly, cancer is usually a mutation. We have killer cells that circulate in our body. So right now, you and I have about 30 billion T cells, a good proportion of
Yes.
which are, right at this very moment in our blood, 30
Right.
billion running around doing their job. And some are helper and some are killer. And then there's also one called the natural killer cell. Now, these have the job of recognizing everything in your body all day long, friend or foe. Is there a pathogen there? Is that a friend or a foe? Do I need to trigger the macrophage to gobble it up? Or is there an atypical cell? Okay, that has the wrong surface markers. I'm going to poke a hole and throw a little hand grenade in and blow it up. Well, what happens after the shot is many of these cohorts of T cells that we have, I call them kind of the Marines of your immune system, your front line. And it includes your T cells, your Pac-Man cells, your macrophages, and your dendritic cells and other organs in your skin, et cetera. And so they work in concert to constantly surveil and clear the body of pathogens or atypical cells. After the shot, there's suppression of many of these cell lines. And when we look at the pseudouridine in these mRNA shots, it's not natural. So this little string of pearls, this mRNA that gets coated in a lipid nanoparticle and put into the body in the shot, that evades the immune system. And that's why they put the pseudouridine in it, so your body doesn't recognize it. So it causes an immune suppression initially. And then the shot goes into your arm, hijacks your cells, and makes your cells the spike factory. Now you have a spike. Now here's what's interesting as well. That spike breaks off of the surface of your cell and goes into circulation as well. So now instead of just staying in the arm, now you have circulating spike in the body. So number one, it's suppressing some of these T cell lines. Number two, the lipid nanoparticle itself will cause a change in inflammation. And then there's studies that show that the pseudouridine can cause decrease in certain protein kinase pathways and certain retinoic acid receptor pathways, many things that are responsible for normal cell function and then can lead to mutagenesis as well. And here's the concerning aspect of this is our body, our cells are making proteins all day long. First, they need that message from our own natural mRNAs, which after giving messages will break down in either a few minutes to several hours. Well, this synthetic sequence is staying in the body. A study at Stanford by Dr. Roltgen et al. Shows up to 60 days later, the synthetic mRNA is still in lymph nodes and spike is still circulating. Now they stopped at 60 days so they could publish their findings. But the question becomes, how many studies are there showing when the body finally breaks down that synthetic sequence? And there's no good study yet. There's essentially zero in the literature showing when the body stops making spike and when that synthetic sequence goes away. So these are suppressing immune cells. And then the other important mechanism, so we have receptors in our body called toll-like receptors, kind of like a toll road. And number 7 and 8, these little pattern receptors tell other cells what to do and say, okay, you're going to fight off viruses. So number 7 and 8 say, okay, keep all these viral families in check. Well, these toll-like receptors are down-regulated after the shot. Great study out of the Netherlands by Dr. Fosse that showed this. Concerningly, toll-like receptors number 3 and 4 were also downregulated. And when 3 and 4 are downregulated, there are multiple cancers associated with a dropout. If we stain in the laboratory these different cancers, you'll see a dropout of certain receptors. And these toll-like receptors are downregulated in aggressive breast cancers. You see them in certain prostate cancers, certain blood cancers, leukemias, et cetera. So this is the tip of the iceberg. And then there's microRNA array disruption, there are G protein disruptions. There's so many things, so many mechanisms that the synthetic sequence and these lipid nanoparticles are triggering from a scientific point of view that could explain why we're seeing this uptick and why they're behaving differently.